Hi students, in this video I am going to show you the detailly the differentiation, dedifferentiation, redifferentiation, which is a confusion topic to the majority of the students, and questions may be asked from this subheading. If you listen properly and see the video, you can give any type of the question in the NEET. Now, before knowing the differentiation, dedifferentiation, redifferentiation, we are going to see what he has given in the root. In the morphology, starting only he has given. That is the root is like this. That is the root cap. That is root apical meristem and a region of elongation, a region of maturation. Three types zones are present. In these three zones, what are present, if you know properly, it is easy to understand this one. That is, the root apical meristem, it is continuously dividing capacity cells are present. They are the cause for the primary growth that you studied already. And reason of elongation. Reason of the elongation means here already he has given in the earlier subchapters. Reason of the elongation, now when the root apical meristem is dividing and the cells are dividing, again the cells will be grown in the two ways, that is the arithmetic and geometric type, that also we know. So in the arithmetic way, they used to send out, for every division they will send out one cell from the cell cycle. That cells used to are called as permanent cells, which used to send out this side. Permanent cells are arising by the arithmetic growth. This is also important bit. And the arithmetic growth, the cells, permanent cells are sending to the proximal side of the root. Whenever it is sending, those cells will be vacuolization. That also term he has given in the starting. Means they will increase they will get the vacuole and increases of size 8 to 9, 8 times to 10 times, means 80 to 90% of the cell is occupied by the vacuole. Such a type of the vacuole used to form in the elongation zone, and so all the cells which are present here are the undifferentiated cells. They are the permanent cells which arise from the root apical meristem. Now, when it is growing like this region will go to the proximal side, and this is the region of maturation will be taken. Region of the maturation, what we discussed in the previous videos, that is, they are these undifferentiated cells which are present in the region of elongation will be differentiated as epidermis, cortex, steel, in the steel again xylem and phloem, like that different types of the tissues used to form. If you understand this one, this already it came, differentiation. What is the differentiation? The cells which used to arise from the meristematic tissue which are present in the root apex and shoot apex will give rise the permanent cells. These permanent cells will be modified as different types of the tissues, and it means that group of the cells is to call forms the cell tissue. So these cells, whatever elongation the region cells are there, they will form the groups of the cells which are the tissues. Those tissues already studied in anatomy: epidermal tissue, cortex, steel, and xylem, phloem, all these things. Like that it is to form. This is called as differentiation. Before differentiation, that cells are there, yes. From where it is arise, he can ask. They are arise from the G0 stage, from the arithmetic growth of the root apex, shoot apex. Remember it. How many questions can be created in this one? You understand. You try to understand. So when they are pushing the permanent cells towards the proximal side, they will be converted into the different kinds of the tissues. That only is telling root, shoot, apical meristems and cambium cells are meristematic cells. They produce permanent cells which differentiate as different tissues at maturity. Maturity is there only. Over, no? That is the, if you want, you note down these notes also and prepare notes on this one. You can give answer, you can understand, you can explain to your friends also. So, differentiation, tissues are maturity. And how it will be differentiated, that also it came. They will be in the differentiated. How these are the cells, undifferentiated cells. They are converting into the differentiated cells by protoplasm and cell wall restructuring. Protoplasm will be that only. It happens structural changes and cell wall and structural means where the cells are, the structure, epidermis it is present on the surface. So structurally that changes in the cell wall. Cell wall means here parenchyma, collenchyma, sclerenchyma, different types of the tissues are present. They are having different kinds of the cell walls. But unfortunately, it is not in their present syllabus. Remember it. So, cell wall, if it is very thick cell wall, protoplasm change. That only he is explaining here in the text. That is, protoplasm secondary and forms a. This is the important bit in this. Protoplasm is absent in the secondary cell wall forms. Lignocellulose. Where in the trachary elements, he mentioned that one in the, what is trachary element? That you will get the doubt. Trachary element is nothing. It is the previous, that is the xylem and phloem, whatever. Xylem is called as trachary element. In that one, four types of the kinds of the cells, tissues used to present. Tracheids, trachea, phloem xylem parenchyma, and xylem fiber. That is a different one. Whatever the tracheids and xylem vessels together is called as trachary elements. Remember it. That you write it if you want. Now the thing is, other examples also you can ask. That is the different one. It is not necessary because it is out of syllabus. Now protoplasm and secondary cell wall formation. Why he has given the trachary element is, the trachary elements are the dead cells. They are not having the protoplasm. Protoplasm is a option. Cell wall will become very thick. Secondary cell wall formation will be taken place. And the secondary cell wall is made up of lignocellulose, important for the need, because textbook has given lignin and cellulose containing secondary cell wall used to form in trachary element. That is the question. You see how many questions can be arise. Now, next it is differentiation. You came to know how it is different. Now, why it is differentiating? Because of its work. The trachary elements has to they are useful for the conduction of the water from the root to the shoot stem. The height of the stem may be the 330 feet also in eucalyptus and upper. That much height it has to transport with high tension. So to tolerate the tension, such type of the mechanism, that lignocellulose will be useful. That is the thing. And the next will be differentiated cells. So these are the differentiated cells. Okay, now the differentiation is over. The primary growth comes. Now, whenever we are seeing in the dicots, they used to, for example, neem tree, 1 year this much size, 2 years this size, like that it is to grow its width. So, whenever it is going the width, growth of the plant, what happens? New cells are arising inside. How it will come? Because they are present in the region of maturation. The cells are not having the dividing capacity. The cells lost the dividing capacity when they came out from the G0 stage. So all these cells are not having any dividing capacity. So differentiated cells also are not having the dividing capacity. But it become need. It is a need. Needed to increase the growth of the plant. Why? To supply growing shoot system more water and more transport of the more food material. New tracks are required. For that one, secondary growth will be taken place. So secondary growth means other xylem and phloem, new xylem and phloem has to be synthesized with the new cells. But the cells are not having the dividing cell capacity. So some of the cells, that is mainly parenchyma cells, living cells, will regain, they will get the capacity of the cell division and convert into the meristem. That is called as dedifferentiation. That one he has written. Differentiated cells based on need, region, cell division capacity regain. They will get the power of the cell division capacity, become meristematic. That we can show here. Here these are the vascular bundles. Phloem is outside and xylem is inside. So already studied in the dicot stem internal structure. Whenever we are seeing this one, it used to present like this. And here one layer he has given and he says that is the cambium. And one more he has given. If you, they are present in the form of the ring. So this is the cambium primary meristem. It is already it is present. It is the phloem. It is the xylem. So in between the xylem and phloem, already cambium is present. Cambium is nothing. It is a meristematic tissue. Remember it. Cambium is present. Now to get the new xylem and phloem, this cambium will become activate and it used to produce towards inside secondary xylem and produces towards outside phloem, secondary phloem. But before that one, it used to become ring formation in between whatever is there. It used to form. It will be continue like this. What the cells are present in between xylem, two vascular bundles? These are the medullary rays. See that one. The medullary rays, whatever the parenchyma is there, that parenchyma which is present in between the two cambiums present in the vascular bundles will be converted as meristematic and it will form a ring. This entire ring is called as cambial ring. Cambial ring is formed. That is the interfascicular cambium he mentioned. This is the interfascicular cambium. That is, this is the interfascicular cambium. Interfascicular cambium. This is already present but newly formed. So the cells which are present in the medullary ray, they regain the capacity of the cell division and become meristematic and forms a continuous meristematic ring in between the vascular bundles. And this is called as dedifferentiation. Already these are the cells, parenchyma cells, which are differentiated and came in this one. They are differentiated. Again, they are getting the regaining of the cell division capacity. So that is called as, again it is modifying. That is called as dedifferentiation. Again different. How it is dedifferentiating? As converting into the meristem. Not only in the interfascicular cambium, whenever it is producing towards outside phloem, towards inside xylem, it used to give pressure on the epidermis. It will be damaged. To protect that one, the cortex upper layers, whatever are there here, they will be converted into the meristematic. Epidermis, cortex, endodermis, and this entire thing is a steel vascular bundle. So outer layers of the cortex will be converted, de-differentiated as the meristematic, and they used to form cork cambial ring. They will become again meristematic. They will produce towards outside cork and inside secondary cortex. That only he has given secondary, that he has not given these things detailedly, but I am giving, remember it. So cork and this cork secondary cortex will be formed from the cork cambial ring. Cork cambial ring is formed the outer layers of the cortex, which is the parenchyma, and they will be they will gain the cell dividing capacity and become meristematic, and cambial ring is formed, cork cambial ring. So this will be producing the cells towards outside and inside. Towards inside, whatever is coming, this is the secondary xylem will come, secondary phloem will come. Here outside the cork will produce, and inside secondary cortex will be produced. Now it is the meristematic. Okay. When it is the meristematic, it also will send some of the cells from the G0 stage out. Permanent cells will be produced by these cambial rings. Those permanent cells again will pick up, forms the tissues. What are the tissue? Cork tissue, secondary cortex tissue, and here secondary xylem and secondary phloem it is to form. So first it is differentiated in this zone, and again it is de-differentiated as the cell dividing capacity as the meristematic cells as the cambium, that is the interfascicular cambium and cork cambium, which are converted from the parenchyma, living tissue. Now you see here, whenever it is forming new cells, again it is coming and forming the region of maturation, like that. So that already differentiated once, again differentiated, de-differentiation, that is as the and regain the cell dividing capacity. Again after the cell dividing capacity, cambium again produces the permanent cells which can be converted into the tissues. So the tissues which are arised from the de-differentiated cells are called as redifferentiation. First differentiation from the apical meristem, and second differentiation will come from the cambium, which is de-differentiated. Now you might have understand. Differentiation means first it is differentiated. De-differentiation, it gain the capacity of the cell division. Redifferentiated, it produced here secondary xylem towards this side, secondary xylem and secondary phloem, and towards this side secondary xylem. Secondary xylem and secondary phloem are redifferentiated, and cambial, whatever the cambium, interfascicular cambium and cork cambium are de-differentiated. Differentiated are the thing whatever we are seeing, that is the epidermis, cortex, steels. In the steel, whatever the vascular bundles, pith and everything. Now here he has given, that only he is telling, this happened because the structural changes of the cell wall and protoplasm, that is one. Protoplasm is absent and secondary cell wall forms lignocellulose, that is the, in the example he has given, what is the trachary element. Differentiation cells based on the need, again they will get the regain cell division capacity, become meristematic. That is the cambial rings. And examples are the differentiation the produce permanent cells. Secondary xylem, this is not given in the book, but it is better to remember it. Secondary xylem, secondary phloem, and cork, and next secondary cortex. They will be produced by the cork cambium and vascular cambium. Plant growth, now it is a where which in secondary growth will be taken place in only dicot plants. That is the bit has given here. So this is the formation of the differentiation, dedifferentiation and redifferentiation can be seen in the dicot woody plants. He has given very clearly. That is also neat bit for you. And woody dicots. This is called as redifferentiation. Redifferentiation, which are the following are the redifferentiated tissue, you can ask. Now the thing is, one more thing is has given in the last paragraph, that is the plant growth, growth and differentiation are open type. In the plant growth, you studied apical meristem, and they are the giving rise new cells. Are always adding the cells to the tissues. Means here this is they are producing the cells and adding to the plant body here. So always it is adding throughout. That is called as indeterminate growth. Indeterminate growth. So that is called as open type. Already studied. Now differentiation is also open type. He's telling. Differentiation is open type means what? Whenever the cells are coming, naturally they are converting into they will be differentiated as a different tissues. That is the thing. Nothing it is. He has given growth and differentiation are open type. If we give the statement, that statement is correct, because as long as the cells means lifelong it is to come, the cells are arising and crossing the region of elongation and entering into the region of maturation, and differentiation will be taken place. So direct continuously adding in the differentiation also. For that only depend upon the location also is given. Here epidermal cells are present in the root cap. Epidermis of this root tip is called as root cap. But one that epidermal cells which are present in this area, that is called as, we are calling it epidermis, means it depend upon the location, it can be given different name. That also he has given in the textbook, remember it. Epidermal cells is to form root cap here. Epidermis is to form the root hair and other things in the maturation zone. So depend upon the location they can be. And here why I shown these two lines, in the location whatever the cells are going in this way, in between this green and black, they will be converted into the cortex. And the cells in between the two green lines, they will be converted into the steel. Means that is the vascular bundles and all the things are present. So depend upon the location, the cells which are present in the elongation zone, they will be converted into the other tissues. That only is telling. I think you might have got perfect idea. Now once more again I will tell. You understand, try to understand this one. This is apical meristem, elongate, that is the, we used to call it as a root apical meristem, shoot apical meristem also same. These two. So it will give rise new cells which are sent out by the G0 stage of the cell division, and this will be followed by the root cap and shoot cap by arithmetic growth. That also he will give, by the which growth it will give arithmetic growth. They used to always add new cells, permanent cells. These permanent cells will be sent towards the root side, and they will be converted into permanent tissue. That is called as differentiation. Permanent tissue means epidermis, cortex, steel. That in that one different job. That tissue will be present. That is the parenchyma, collenchyma, sclerenchyma, and xylem, phloem. So many types of the tissues. It will be differentiated. That I discussed clearly in the previous video. Kindly see that video. And this is the differentiation. Without differentiation, what happens? You will ask. What you will tell? Without differentiation, plant cannot survive because different tissues and the structures are required for its survival. The huge plant, well, flowering plant, and all the big plants. So differentiation is must. And now the differentiation will be the tracker elements example he has given. Tracker elements are nothing. Xylem, the xylem will present xylem element, tracheids and xylem vessels. Both are the having the hollow inside. Means that elements, what they will do, they will conduct the water. With a high tension, the water used to means that much tolerating capacity that will have. So the cells which are differentiated, yeah, the tracker elements, they won't have the protoplasm. This is one more bit. And secondary cell wall is made up of the lignocellulose. This is one more bit. And next is the differentiated cells needed. Whenever the need coming, they will regain the cell division capacity. Cell division capacity they will get. So already differentiated, again it is getting dividing capacity like this. And this, whatever the cork cambium. So whatever they are regaining as the dividing capacity and become meristematic, that is the cambium. So the cambium is the de-differentiated, from which one the here whatever the medullary rays made up of parenchyma, cortex is also made up of parenchyma. So parenchyma cells will regain the cell dividing capacity and become meristematic, and it will produce new permanent cells which will be converted as the tissues. So de-differentiation is present in all plants. If you give the wrong, it is statement is wrong. It is present only in the dicot. Dicot plants only will increase the width of the stem. It is not in the monocot. Coconut tree, you see, if it is this much size in the right from the beginning to ending, it will be that much only. Toddy palm, like that only. All the palm trees, which are the monocot. But if the tamarind tree, neem tree, it used to increase. Banyan tree, they are the dicot, woody plants. So de-differentiation and redifferentiation will be applied to woody dicot plants. Remember it. This is also important bit. So in this way, they used to produce secondary xylem and phloem. So they are producing the new tissues, permanent tissues. So that permanent tissues again it is forming from the de-differentiated tissue is called as redifferentiated tissue over. And next is in the last paragraph he has given that the growth and differentiation are open type. That also I explained. So try to understand and listen to this one, and the diagrams also you see by zooming, and you will understand. Prepare the notes. You never get any doubt up to the NEET examination you give. It is enough. If you like this video, please give the like mark. And such type of videos, so many are coming. Already I made up to now 17 videos, long videos, and 10 short videos I made in the photosynthesis, respiration, and growth also I am doing. It will be completed by the with 3 videos more. So, and I am going to do more videos, nearly 70 to 80 videos will come. 80 or 90 also, up to 100 it will come. Every week 2-3 will come, and 2-3 short videos will come. All these videos contain such type of information. If you want to see those information and if you want to prepare properly for the NEET exam, this information is very, very important. Some of the points you won't get in any books also. Means the points are not new, not out of syllabus. They are within the syllabus. I am showing you in the different ways. So this is, I am just initiating. Your brain also will develop to, if any creation comes, it will understand. Means concept orientation I am giving. If you want to see all those videos, please subscribe this channel and timely you see the whatever videos I am releasing. Thank you.